Hello class. So this is a continuation of our discussion on part 2 of your microbial growth um, requirements. So last video, we have discussed the different factors affecting microbial growth rate. Now we will review microbial growth curve. So this figure here graphically represents the number of living cells in a microbial population over time. So the y-axis is the log of cell numbers, which can be in the units of CFU or colony forming units per milliliter or per gram. And then you have here your x-axis, which is time. So this graph here will show or predict a pattern between the growth of your microorganisms that are grown in a culture. So now let's discuss the different phases of your microbial growth curve. So the first phase of the microbial growth curve, we have here the log phase. So the log phase is the initial phase of your microbial growth, which is characterized by cellular activity, but there is no growth yet. So during the log phase, there is only cellular activity. So a small group of cells are placed in a nutrient-rich medium that allows them to synthesize protein and other molecules necessary for replication. So during this phase, the microbial cells will increase in size, but no cell division occurs in the phase. Okay, so I hope you can remember that. Again, during the log phase, it is the initial phase where there is only cellular activity, where the cells just increase in size, but there is no cellular division occurring on the log phase. So moving on, increasing the time, you have microorganisms will continue to the next phase, which is the exponential or log phase. So after the log phase, after this phase, bacterial cells, they can enter into the exponential or what you call here the log phase. So during this time, it is where the cells will divide through binary fission. So we have discussed on our chapter 1 the binary fission or the doubling of your bacteria in numbers after each generation time. So during the log phase or the exponential log phase, the microbial cells, for example, for your bacteria, they will divide or they multiply or double through your binary fission. So different metabolic activity such as your DNA, RNA, as well as cell wall components and other substances necessary for growth are generated for division. So it is also during this phase that antibiotics as well as disinfectants are most effective, as these substances are typically target bacterial cell walls or the protein synthesis processes of DNA transcription as well as RNA translation. So again, during the exponential log phase, so the bacterial cells or microbial cells, it will double in numbers through either binary fission or through meiosis or mitosis based on its generation time. So different metabolic activity in terms of the different necessary requirements, different substances necessary for their requirements will be generated during this phase. Okay? So further extending the time, we have here the stationary phase. So eventually, at during the stationary phase, the population growth experience in the log phase will begin to decline as the available nutrients will become depleted and waste products start to accumulate. So bacterial cell growth reaches its plateau. So you have here a plateau or a stationary phase where the number of dividing cells equals the number of dying cells. So this results in no overall population growth. So again, during the stationary phase, there is no overall population growth because um, the number of your dividing cells as well as the number of dying cells are equal, or the cell growth already reaches a plateau or a stationary phase. So under the less favorable conditions, um, competition for nutrients will increase during this phase, and the cells will become less metabolically active. Okay, so spore forming bacteria, they will produce endospores in this phase, and pathogenic bacteria will begin to generate substances that help themselves survive harsh conditions and consequently cause diseases. So again, during the stationary phase, the population growth experience during your log phase will start to decline as the available nutrients for your microorganism to grow will be depleted, okay, and waste product will also start to accumulate. So during this phase, there is um, the bacterial cell growth already reaches a plateau. So you have here the peak phase where the number of your dividing cells will be equal to the number of your dying cells. Then further extension of the time, you have here the death phase. So since, as discussed earlier, during the stationary phase, there is already a decline in the nutrients. So as the nutrients become less available for your microorganisms and waste products increases, the number of dying cells will continue to rise. That is why during this phase, the number of living cells or the number of your log cell numbers in CFU will will decrease exponentially, and the population growth will experience a sharp decline. So you have here an exponential decrease of your number of living cells, as well as the population growth will also have a sharp decline. So as dying cells lyase or break open, they will spill their contents into environment, making these nutrients available to other bacteria. So this helps spore producing bacteria to survive long enough for spore production, okay, which can support the life of other bacteria. So again, during the microbial growth curve, during the log phase, it is where most bacteria or most microorganisms only has cellular activity in which the cells increase in size, but there is no cellular division that is happening. So it is where during the exponential or log phase, where there is continuous dividing of your bacteria through binary fission, or your molds through meiosis or mitosis, and it is during the exponential log phase where there is a doubling in number of your microorganism, and further increasing the time, you can observe there is a plateau or a stationary phase where the number of microorganisms, the living microorganism, is equal to your dying microorganism. So during this time, basically, all available nutrients will be depleted. So nagkahinay-hinay na sila pag reduce or decline sa stationary phase, since most of the microorganisms um, will utilize the available nutrients. So, further extension or further exposure to longer time, you have here the death phase, where there is the number of your living cells. Okay, as you can observe here, the number of your living cells basically decrease exponentially, as well as there is decline in the population growth. So that is why, for the longer the time, you can also observe that the microorganisms will basically decline in number. So this is just a review of the microbial growth curve. Again, we have here your log phase, then your log or exponential phase, and then your stationary phase, as well as the death phase. So now let's discuss the different kinetics of microbial growth. So unicellular microorganisms, such as your bacteria, they mostly replicate by a sexual process or through binary fission. So bacteria, each bacterial cell will grow to full size, and then it replicates its genetic material, then divides one cell into two identical daughter cells. So by identical means, two cells will divide into four, and then four cells will divide into eight, and so on, leading to an exponential increase in the cell number of your microorganism. So this explanation will this kinetics of your microbial growth explain why there is an exponential rate for your microorganism, because there is an exponential increase in the number, since most of the bacteria divides through binary fission. So again, one cell can divide into two, the two cells can divide into four. So as you can see, there is an exponential increase in the cell numbers. So now you are asking, why is there exponential curve for your microbial growth curve? So now let's see this figure here, which plots the number of cells in population against time. So the x-axis is the time, and then you have here the number of your microorganisms. So this is in log form. So just like this one, this is the log of cell numbers as well as time. So this one also, this is the log of your microbial cell per time. So as you can see here, as the time increases, you can observe that there is exponential increase. So after 20 minutes, the number of your microorganisms is 500. Then after 40 minutes, the number of the surviving cells is 1,000. Then you have here 2,000 after 60 minutes, and then you have for 70 minutes you have again greater than 2,000. So as you can see, there is a exponential curve where your microorganisms will divide exponentially. So I have discussed earlier that unicellular microorganisms, such as your bacteria, they grow in binary fission. That is why you have exponential growth in your number. So under favorable conditions, most cells occurs every 20 minutes. So when all factors are favorable to your microorganisms, most cell division, or through, for example, for your bacteria, which it can have binary fission, so cell division will occur every 20 minutes. So I hope you understand why there is an exponential curve for your microbial growth of your microorganisms. So we also have here a plot of the log cell against time during exponential growth. So you can observe here that when there is exponential curve, you can also plot a straight line following the log cell number as well as time. So this one, you can basically form a straight line from this exponential curve. Okay? Sorry. So, this part here, basically, pwede mo na siya masulatan line dire. Okay, this part. Okay, so there is an exponential uh, straight line that you can plot for your um, cellular number versus your time. So again, we have here the microbial growth rate. So again, we have here the log phase where there is only cellular activity, and during the exponential log phase, you can observe that there is basically a straight line for this one since your microorganisms increase exponentially. So for 20 minutes, they can increase by 1,000 or by 5 by 500 or in log. So this increase here is 1 log. This increase here is 1 log. So there is basically an increase of your log cell numbers per 20 minutes, as you can observe in this plot here. So there is basically, this is 1 log cycle, this is 1 log cycle as well, and this one is 1 log cycle. So that is why your log phase is exponential log phase. So there is an exponential increase in the number of your microbial cell. So under optimal conditions, the population of your cells will double in a constant and predictable length of time known as the generation or doubling time. So again, under normal condition where the temperature, the pH, as well as the oxygen is optimal, the growth of your bacteria will double through binary fission in a constant as well as predictable time. So this is what you call generation time. So the value for a widely used laboratory bacterium is your Escherichia coli is 20 minutes. And for most organisms, it is less than an hour. It is less than an hour. So there are some bacteria, however, whose generation time is many hours. Thus, during exponential growth, the number of cells can be expressed as NT equals N0 times 2N. So where NT is the total number of cells, and you have here in N0, which is the number of cells at the start of the exponential growth. So this is your N0 is the initial number of your cells, and then we have N, which is the number of doubling times that have elapse. So the um, most of your E. coli, the generation time is 20 minutes. However, for those other microorganisms, you can express the number of cells through this formula here. So NT is equals to N0 times 2N. So the study of pure cultures made it possible to determine the properties of a specific organism, such as its metabolic characteristics or its ability to cause a particular disease. It also opened up the possibility of classifying microorganisms on the basis of the characteristics they display in the pure culture. So again, when you cultivate microorganisms, they have specific properties as well as they can it is possible to classify microorganism based on those characteristics that are displayed in the pure culture. So when you grow microorganisms, there is incubation time as well as an optimal temperature for your microorganism to grow. So it needs time as well as the optimum temperature for your microorganisms. So now we will discuss how can you cultivate microorganisms or the how can you grow microorganisms in the laboratory. So the artificial culture of any organisms requires a supply of the necessary nutrients. So your microorganisms will basically not grow if they don't have any supply of nutrients. So together with the provision of appropriate conditions, such as the different factors that I've discussed earlier, so such as your temperature, pH, as well as oxygen concentration, this will help the microorganisms to grow. So the nutrients and conditions provided in the laboratory are usually a reflection of those found in the organism's natural habitat. So it is also essential that appropriate steps are taken to avoid contamination. So to cultivate your microorganisms, basically what you need for your microorganism to grow is basically their nutrients. So these nutrients can be coming from your food. Okay? So most foods, again, they contain water, protein, okay, fat. So these nutrients are basically required for your microorganisms to grow. So as well, in addition to the necessary requirements coming from the food, microorganisms also require a specific pH for them to grow, as well as incubation time. So how long you will let the microorganism grow, as well as an optimal temperature for their growth. So your mesophile basically has different optimal temperature compared to your psychrophilic microorganism. So your mesophile will basically not grow at lower temperature, and uh, it will not grow at higher temperature than its optimal temperature. So apart from temperature, most microorganisms can or cannot or will not require oxygen to grow. So you have anaerobic microorganisms which don't require oxygen. Others are aerobic. Then apart from that, microorganisms will require moisture for them to grow. So this um, different factors here are needed for you to cultivate different microorganisms. So again, memorize FATOM. So food, acidity, time, temperature, oxygen, as well as moisture. So before discussing on how to obtain a pure culture, we will end on this part here on how to cultivate microorganisms, because this will be needed for your experiment number one. So uh, we will be using different culture media. So the culture media will serve as a food for your microorganisms. So it contains the necessary nutrients for your microorganisms to grow. And then we will decide on the time, as well as the temperature, as well as the oxygen and acidity for your culture media for different microorganisms to grow. So for the first experiment, we will only prepare the culture media, but later on, we will basically grow microorganisms. So this is the end of our discussion on chapter 2. Then next, for chapter, sorry, this is chapter 3. So for chapter 4, we will discuss how to obtain a pure culture, as well as the different methods for your microbial, on how can you obtain a pure culture. So let's end in this slide here. So that is all for this video. Thank you.